Next up, we have Dr. Nicholas Somm. Currently, that is a lot louder than earlier. So, okay, sorry, everyone. He is currently the unit leader of the USGS California Cooperative Fish and Wildlife Research Unit at Humboldt. He joined the cooperative unit in November of 2023 as a unit leader. He also teaches graduate courses on statistical methods common to fish and wildlife projects, such as generalized linear models, applied Bayesian modeling in the Department of Fisheries Biology. And I believe he's been doing that for a number of years prior to accepting that position. His educational history includes an undergraduate in mathematics from Regis University, a master's in statistics from University of Washington State University, and he got his Ph.D. from Oregon State University, where he studied time series and spatial statistics for data originating from streams and river networks. So prior to joining the Cooperative Research Unit, he was a statistician and program lead for the Fish and Aquatic Conservation Program at the Arcata Fish and Wildlife Office. His research focuses on answering ecological questions with quantitative tools aimed to inform resource management decision makers. His main interests include water management and the ecology of Pacific salmon, and his work includes both methods development and applied science projects. His applied work has ranged from the Columbia River to the Sacramento River, but is focused primarily, or for the most part, on the Klamath River Basin. And he frequently works on models for aquatic habitat, population dynamics, and disease dynamics. And everybody in the program knows that Nicholas has worked for us, done work for the program for a number of years, and has been a key player in developing several models that we use on a regular basis to help better inform management. So with that, I'll give it up to Nick.
All right, thanks, Ken. It's super fun to be here. The Trinity is one of my favorite spots as a river. And as he mentioned, I've worked for a long time with the program, so it's fun to see some of the presentations today and the next couple days. I was asked to talk about Sea Shasta. And so the approach I've taken is a combo of talks we've seen today. The timeline of the science of Klamath River Sea Shasta is actually pretty similar with the restoration program. So I'm going to talk about some kind of lessons learned over 2 decades of work, certainly a lot of which happened before I got here. And then I'll talk about a little more specific results and some technical things, including finishing with some ideas about Sea Shasta risk to Trinity fish. I mentioned a lot of this work started before I got here. I put up a list of some OG names of people who really drove the Sea Shasta work. And already, I know I left some people off, including Wade Sinnen, who isn't here, but worked with CDF and W in the basin for a long, long time and certainly contributed to that body of work. And so the title, Monitoring to Science to Management, really is about how the importance of these monitoring programs is in helping drive science projects and then turning those science projects into management informing tools. So I'm happy to say this is the first time I've put up a map of the Klamath River and I don't have dams on it. So they're not totally down yet, but it was kind of fun to be in a position to do that for this talk. And those 3 stars represent locations of rotary screw trap, juvenile outmigration locations. These are projects that have been implemented for decades, partnerships between lots of tribal and federal and state folks involved in monitoring juvenile salmon in the Klamath Basin. And in general, historically, as you would move downstream among these 3 traps, you would tend to catch more fish. We have tribs that are coming in that are contributing fish. We have main stem spawners that are contributing fish. And although, of course, there's some mortality, over time, generally, you would pick up more fish as you move downstream. Well, starting in the early 2000s, that started to change, where we would find in our traps, and this, again, I'm using the collective we here, they'd catch fewer fish as you move downstream. And not only that, they would catch dead fish or fish that looked like you see the pictures here with these distended bellies, and people got really, really concerned. They didn't exactly know what was going on, and having fish mortality at this scale was certainly troubling for management and people concerned about the status of these populations. And so this really set out a course of work to discover what the problem was and then eventually try to manage for it. Okay, and that's really what I'm going to spend a fair amount of time talking about here. So we had these monitoring programs, like I said. We had the juvenile out-migration monitoring, which identified that we had this problem in the first place. We also had spawning monitoring going at that time, which proved to be super important as well. But what it required was something that seems to be an even more and more challenging idea in the last decade for sure, is it required extra monitoring. It seems to get more and more difficult to find funding to add new monitoring, even if there seems to be a real science need for it. But a group of people were able to do that. And I said here on that slide, a lot of partners, and this really was a massive collective effort. I mentioned we had federal agencies working on the Klamath in conjunction with tribal partners. We had the state agencies. We had research institutions like Humboldt and Oregon State University. And it took these group of people really working together to pull a lot of this off. And so where did that start? Well, there was thoughts that it could be a myxozoan. It could be a parasite. They weren't exactly sure what it was or how it was working. But that involved adding water sampling, which continues to this day and is a critical part of water management on the Klamath. And so, yes, they were collecting fish and weighing fish and counting fish, but they certainly had to add a new component to that monitoring, and that involved collecting fish on a weekly basis that were then sampled internally to look for signs of infection if it wasn't obvious on the outside, and through a lot of histology and other laboratory means to detect if the fish were infected, how infected were they, and to really start getting an idea of how prevalent this was across the population. That's for juveniles, and that certainly was the case also for adults. Additionally, they had to implement experiments. Again, they weren't exactly sure what was going on, but they knew obviously they're fish, right? So they're getting sick in the water. So they implemented what are called sentinel exposures, where they added fish to these cages, and they would put these cages in the water for 3 days and see if they could observe infection. Now, sea shasta infection doesn't reveal itself over the course of 3 days. So they would take these fish back to the lab and rear them for a couple months and track what was going on. And really, again, with this idea of a myxozoan, there was a life cycle to think about. The life cycle of these parasites often includes not just a fish host, but an intermediate or alternate host that each are releasing these infectious agents. So the water sampling, trying to figure out if they can detect these infectious agents, and the monitoring to try to discover if they could figure out what the other host might be. And then another super important part of this that I would say, having had a lot of conversations in particular with Nick Hetrick over the years, who was at the start of this, was they really had to focus on communication. And they did that for a couple reasons. One is, I just talked about how hard it is to fund new monitoring or funding new science projects. This was adding a lot of science expense on top of an already well-funded monitoring program. And so they really needed to stir up public support and agency support to carve out the funding to implement a lot of these. And they had this idea for a fish health workshop, which continues to this day, actually is going to happen next month. And the impetus for that fish health workshop was really to get the press there. It was one thing for scientists to talk to each other, and they were doing an okay job of doing that. But they needed to do a better job communicating with the public and getting the press there to help spread this word that was really a strategy, again, to try to stir up some funding. And that did lead to the discovery of the life cycle. Okay, and so it turns out that this parasite alternates with a benthic invertebrate worm, now called an annelid. Okay, and those annelids release actinospores into the water. Those actinospores will only infect fish. The fish become infected, and they release a myxospore that will only infect the annelids. And I put general up here because this is the general thought about the life cycle and likely how it plays out in most years, where the adults return in the fall, some get sick, some release spores. Juveniles pick up the spores the annelids release in the spring, some get sick and release spores, and so on. We also think, though, in years where disease is really hot, we might get a double cycle in the spring where juvenile fish get sick and release spores that are then flipped again. And so, again, this is just in general. A couple of other key pieces of information that came out of those early studies is that C. shasta is endemic to the system. It existed for years and years, or well, millennia, in balance, right? But things that happened to the ecosystem that had caused that relationship to become out of balance. And certainly what they observed now that they were keeping track was some years were much worse than others. Some years there was a tremendous amount of what appeared to be disease-induced mortality, and some years where there was very little. So phase 2 is about where I came into the system. Now that there was a good idea about the life cycle, we started asking other kinds of questions. Can we quantify and predict the impacts? And again, that brought about more collaborative projects and more science inquiries. Of course, we had to keep this monitoring going, but we expanded that even still, where we started looking at if we could describe and model annelid habitat. So what's suitable areas for these annelid worms? And we also tweaked some of the original experiments. So I mentioned those sentinel trials, which were 3-day exposures. We wondered, wild fish, even hatchery fish, they're not in the system for 3 days, is being exposed for longer associated with higher rates of mortality. So we set up these experiments to expand them to longer durations. We started churning out a lot of this work. And in particular, we started having stakeholder meetings, in particular with Klamath Basin water users who are becoming increasingly concerned about the way the conversation was shifting in terms of how water management might be used as a mitigating factor for some of the disease stuff. And so they were invited to these fish health workshops. They were a part of the conversations, and we did our best to try to make sure everybody was informed. And so what are some of the findings that came out of that work? Well, I mentioned that annelid habitat model. The real take home there is that suitable annelid habitat is stable substrates and low velocity areas. Okay, and so what you see are 4 pictures here, and it's a little small, but the upper left is bedrock. Then we go to cobble in the upper right, gravel in the bottom left, and silt and sand in the bottom right. And so a darker color represents higher probability of presence, so more suitable habitat. So we see the darkest colors, of course, in that bedrock, and the lightest colors in general in the sand. And you'll notice the intermediate depth. So depth is on the x-axis here. So you'll see that the darkest colors where they exist are in the middle of that x-axis, okay? And we have this kind of sweet spot of depth where it's darkest, but as you increase in velocity going up the y-axis, the colors tend to get lighter. So we have this intermediate depth, very stable substrate, and low velocity is really what creates the kind of habitat where we find the annelids. And in terms of mortality pressure, what's driving mortality in fish? We have another kind of multi-axis graph here. So on the x-axis here is water temperature, and on the y-axis is spore concentration. So the dose of those actinospores, and we see that as we go up in the y-axis, we lead to increasing probability of mortality. So here, black is the lowest probability of mortality, moving to white, which is the highest probability of mortality. And so again, higher dose, it makes sense. Higher probability of succumbing to infection. It's a little more interesting on the temperature part of this axis, where we see a curvilinear relationship where it gets lighter and lighter as we move in the increasing x direction, and then darker again. And that might seem counterintuitive, and that's not actually a result of the way the disease is playing out in the fish. It's actually that as temperatures get warmer and warmer and warmer, the spores disintegrate. And so you end up with fewer of the spores that are technically available to infect the fish. Okay, and then I put exposure duration of 7 days on this slide rather than present a series of slides on exposure duration. That hypothesis proved to be true. Shorter exposure means less probability of mortality, and a higher exposure duration certainly corresponds to a higher probability of mortality. And that's certainly going to be interesting when we start thinking about potential for Trinity fish in a little bit. Another interesting tidbit that came out of all this work is that the environmental risk itself might help you describe the fate of a group of fish at any given time, but we really were thinking about what's happening to the population, right? Okay, and so in these graphs, we have a dashed line that's going to represent risk. Think of that as sea shasta mortality risk, okay? And a black line that's going to represent population estimates. So both of the x-axes here are weeks throughout a monitoring year. Okay, so you can think about moving from early in the year to later in the year as we move along the x-axis. And so in 2014, you see that we have an initial wave of outmigrants, okay, and then a late wave of increasing sea shasta risk. So in that kind of year, you would say, yeah, disease risk actually got pretty bad, but most of the fish had outmigrated by the time that happened. So that would be a year where at the population level, there wouldn't be that dramatic of an impact. Whereas if you look at a year like 2009, you see over time, the increase in risk almost perfectly coincides with the outmigration timing, right? And so that's a year where we would predict sea shasta is going to have a really big population hit. And then, of course, there's years like 2012, where even if the relative change in disease risk is coincident with the population, the risk is really low, right? So that would be a year where we wouldn't predict that would be that much population impact because disease risk never got very high. Okay, so now that we had some of these key findings, we set out to do really what we wanted to do all along, which is, can we use this information to inform management in ways that improve outcomes for fish? Okay, and really that came down to, can we disrupt parts of this sea shasta life cycle and get this broken a little bit? Okay, so some of the tools we thought about doing that with are annelid habitat. I talked about what kind of conditions are good for annelids. So we wanted to try to minimize low velocity protective zones for them. I'll talk a little bit about that in a minute. And certainly if we could interact with the system in ways to reduce, say, spore concentrations, that would be a benefit to fish health. So again, phase 3 here is really where we're trying to use this information to make a difference. I'll also note here, I won't call this a science finding, it was more of an aha moment, but this actually is the hydrograph of the managed Klamath River at Iron Gate Dam since Iron Gate Dam was put in, okay? And so this is right when the Iron Gate was put in, and you'll notice we've got these color bands, and those color bands just correspond to increasing levels of geomorphic work. So in essence, as a flow gets higher and higher, it's able to move larger substrates and cause more disruption. And the aha moment about looking at this graph is right here is about 2000. And if you'll notice, prior to that point, there was lots of flows in terms of frequency, even in the managed system that got into that yellow and blue. And that's really where we get sediment disruption, particularly some of the sediments that we find the annelids on. But you'll notice after 2000, again, without any intentional change to geomorphic types of flow regimes in the Klamath, it was managed for purely different reasons, we basically lost most of those flows, right? We did get one here about 2010, but clearly we started thinking maybe there is much more of a direct link to geomorphic flows in particular and the annelids. So we started applying these tools in hopes to help, and we think we have been helpful. They've been used in lots of different ways. So for example, here, Bureau of Reclamation asked us to apply these tools, and we have them for both Chinook salmon and coho salmon. they are differentially infected. That's an in-the-weeds detail I could answer questions about later if interested. But we started looking at historical rates of infection and then make predictions with our models under different proposed actions or different kinds of flow management alternatives. And we did that both for kind of standard long-term proposed action development planning and in emergency situations when things like alternate flow plans for droughts had to be implemented. We applied these models to help with analysis that NOAA was conducting as a part of their FERC analysis, as a part of the dams out process. So we definitely had to make some assumptions about what might happen with disease in a dams out environment. But again, our models were included there, and we helped conduct some of those analyses. And the disease has even been implemented as a part of the regulatory framework for take statement evaluation. So both estimated annual prevalence of mortality from sea shasta for Chinook and coho salmon are currently included in the take statement for the Klamath project. And then for the last several years, CDF&W has asked us on a weekly basis to provide relatively real-time predictions of disease risk to inform their hatchery release timing. So as it approaches the situation where their fish are getting large enough that they can be released, they start implementing weekly meetings. Day of, we get current water temperature information, we bring in the most recent spore concentration information, and we make predictions like, you know, we're going to predict that maybe 40% would succumb if you release the fish this week. And then, of course, that's couched with predictions about how future water temperatures might change or the hatchery might run out of room. Again, that's been a decision that's partner-driven, but this has just been one piece of information to help drive a lot of those conversations. And then maybe my favorite slide in the whole slide deck here is one that I never have actually put into one of these talks before, but I think all of us actually leave these slides out, but we were wrong a lot, you know, and we had to own that, and sometimes that dealt with a credibility issue, or sometimes people understood it was a part of just the scientific process, but we had hypotheses that didn't turn out to be true. I presented some, of course, that did. We didn't even name the things right. So it used to be called Ceratamixa shasta. The genetic reasons that's not what it's called are actually way over my head, and I won't get into that, but it's Ceratinova shasta. For years, the benthic invertebrate worms were thought to be polychaetes. They're annelids. Again, that distinction, you'll have to talk to some of my colleagues at Oregon State about why that's important. I've just now recently defaulted to annelid and not made the mistake of calling them polychaetes. So a prime example, we thought that more returning adults in the fall would lead to higher disease risk the following spring. More adults coming back, more adults getting sick, more spores being released into the system to infect their progeny. That actually didn't turn out to be true. And the reason for that is there's a lot of variation in the spores produced by adults. There's actually a very small number of adults, we call them the lucky losers, that produce millions and millions of spores. And the vast majority of adults, that even those that are sick and infected, produce a relatively small amount of spores. The flow events that have largely been adopted into these regulatory planning environments are flows aimed to scour the annelids, and they're called flushing flows. That's the common phrase to describe them. You may have heard about flushing flows. But there's other kind of flows that have been hypothesized to be generally beneficial for disease risk. One of those is dilution flows. And so by dilution, it's exactly that. You increase the volume of the water, so the concentration of the spore drops, and therefore with lower spore concentrations, less infection and eventual mortality. Although we can measure effects of these dilution flows in real time, they don't persist. So if you raise the water to dilute it and then drop it, you return to the same concentration that you had before. And the reason that's important is there's a lot of variation in the spores produced by adults. There's actually a very small number of adults, we call them the lucky losers, that produce millions and millions of spores. And the vast majority of adults, that even those that are sick and infected, produce a relatively small amount of spores. in order to carry out a dilution flow that would have an actual population level effect, you would have to have that water elevated for quite a long time. I mentioned these annelid worms. There was thoughts that it could have been snails or mussels that were the alternate host. And then I mentioned briefly, only adults contribute myxospores. We now know that in some years, the juveniles are getting sick and dying quick enough that they're actually adding myxospores to the system. And like any water in the West issue, and again, Sea Shasta has been a water in the West issue because flow management has been targeted often as one of the remedies. You know, we've had to deal with anger and fear for people who share different values about the use of water. And so one way that we dealt with that is we tested some hypotheses that weren't ours. And in particular, the water users approached us and said, well, we think a perfectly good management alternative would be, instead of more water, it would be, why don't you dry the river out and kill all the annelids and desiccate them? And obviously they didn't mean drain the river completely, and we said, well, we'll look into it. And because we had these habitat models for annelids. And I mentioned earlier the sweet spot for annelids was at an intermediate depth, low velocity stable substrate and intermediate depth. And so the chart you're seeing here is a stacked bar graph where the darkest bottom color is actually the highest quality annelid habitat at a series of discharges, so increasing discharge from left to right. The lighter yellow color is the moderate amount of habitat, and green is basically what's left over, habitat that annelids wouldn't occupy. And so as the river gets drier moving left to right, as we reduced flows, and again, we had to rely on 2D models for this because we couldn't actually drop flows for important reasons. We see an overall decrease, but the only color that's decreasing is the green, the low quality habitat, because we couldn't get to those intermediate depths, which is where all the annelids were. So we were drying up areas that weren't annelid habitat. So what about Trinity fish? How can we think about Sea Shasta risk for Trinity fish? Trinity fish do enter the Klamath. We know they do that, of course, at Wichipec, right here at the confluence there. What kind of information do we have that might inform some impacts to Trinity fish? Well, Bill talked about juvenile outmigration monitoring, and that's at a series of rotary screw traps that are just downstream of Willow Creek, and they're called the Willow Creek Rotary Screw Trap sites. And there's actually a water monitoring station on the main stem Klamath below the confluence with the Trinity River, and I think Chad mentioned Tully Creek. That's actually the lowest water monitoring site on the Klamath main stem to collect spore density samples. And so we can look at those pieces of information to think about what might be going on with Trinity fish. There's some really big caveats. One is that the Tully Creek site is not one of the standard index sites. It was added later, so we have more recent data on water samples at Tully Creek, but not as much as some of the other sites. And based on the currency of the juvenile outmigration estimates, we don't have the Trinity River outmigration estimates in 2018 and on. So I'm going to show you a couple years where we have the concurrent water sampling data and the juvenile outmigration data, which is only 3 slides, 2015, 16, and 17. But I think that gives us actually a good flavor for what this might look like more generally. And then the other big caveat that really pertains to this Tully Creek site is the genetic methods that are used to get the concentrations of these spores in the water cannot distinguish between the spore that infects fish and the spore that infects the annelids. So we can't tell if they're actinospores or myxospores. We can just tell that they're Sea Shasta. And the reason that's important is, again, myxospores only infect annelids. So there's a chance, in particular in years where we are having juvenile fish mortality upstream, that water samples at Tully Creek are picking up actinospores and myxospores, or one or the other. We just can't distinguish. So I just want us to keep that in mind when we look at a few of these slides. And I just throw in there, the only way to confirm that it's an actinospore after you've done a water sample is to actually put caged fish there and see if they get sick or not. Because if the fish get sick, we know it's actinospores. So this figure is similar in style to the ones I showed earlier, where the dashed line can be thought of as disease risk in a time series from early in the season to later in the season, and the solid line is the weekly fish outmigration numbers. And there's actually a data gap here, so the dashed line does have a couple breaks, but I think we can probably fill that in with our eyeballs if we want. So 2015 is a notorious year in the Sea Shasta Klamath world, where even in the upstream monitoring sites where we have long-term data sets, the spore concentrations were over twice as high as we'd ever seen before. Okay, and so the water monitoring down at Tully Creek in that year peaked at 800 spores per liter, which is an astronomical spore concentration. Again, that was a really hot year for juvenile mortality. I would predict that that's a combination of actinospores and mixospores, certainly in that year. But regardless, not all of that would be mixospores. So there definitely was a year of elevated disease risk. That is coincident, almost perfectly overlapping, of course, with the outmigration timing at Willow Creek. Another caveat, of course, is there's some amount of time between Willow Creek and the Tully Creek site. But I'm not so worried about that just for this exercise. The next year, 2016, also looks like another year from the examples before, where we see some elevated disease risk, but later in the year, after it looks like the Trinity fish may have outmigrated. Again, if we shift that a couple weeks to coincide with maybe hitting that Tully Creek site, there might be a little more overlap. And then 2017 is a year where spore or disease risk just never really got anywhere. It stayed low all year long. So we see that dashed line of disease risk staying flat along the x-axis there, even though we have the bouncing outmigration monitoring estimates going along there. So sea shasta is something that, in my opinion, is something that's hampering Trinity origin fish. In my opinion, I think there's probably years where a portion of Trinity fish are being impacted by sea shasta in the lower Klamath. I think it's fewer years than the Klamath mainstem fish because there's just a much higher frequency of their outmigration timing overlapping with disease risk. And further, historically, Tully Creek has been a site registering the lowest disease risk spore concentration values among all sites. So in many years, it's much lower, not in all, but in many years, it's much lower. Again, I think bad years, even Trinity fish are getting hampered by really rough disease conditions in the Klamath. But I'll also note there's some recent experiments based on some work in Davis that suggest that there might be some fairly severe, or drastic, I should say, differential susceptibility impacts to sea shasta by origin. Okay, so there's a chance that genetically, Trinity fish are much more or much less susceptible to sea shasta infection than Klamath fish. Okay, I think that would be something that would be very interesting to test out. There's been some experiments. Sea shasta is also an issue in the Central Valley, and there's some emerging research to suggest that this might be something that we need to investigate, maybe statewide. And I certainly wouldn't make assumptions that Klamath infection rates for a given temperature and spore concentration are going to automatically transfer to fish of different basins. And again, just kind of a higher level piece about what did 20 years of sea shasta work, what led to the successes. The ability to try things and fail and not have that bring the house down was huge. When you're in an environment where you require that there's no failure, that means you're probably not going to learn very much, certainly not as much as if you have the chance to do experiments and to learn from it. Obviously, the extensive collaboration is what really made this possible. Not just in terms of there was so much work to do, but the vast diversity in backgrounds and perspectives and skill sets, and even willingness. And I put bad ones in quotes, kind of in jest. Trying to get larger buy-in by listening and trying ideas, even if they're not your own, even if you don't think they're prone to work. That's not really the point, right? And you're also losing out on potential opportunities to learn if you're not willing to entertain some of those other ideas. All right, there we go. Happily take some questions. Was I that late? Oh, that's funny. I thought I was early. Yeah. Yeah, so Todd, that's what I'm doing. Yeah. So Todd asked if basically what do we predict is going to happen given all the suspended sediment that's been mobilized because of dam removal? Is that a good enough summary? So the prevailing idea was that a lot of the annelids were going to get covered in a thick blanket of sediment, and that perhaps this year disease risk would be much, much lower. I also will say the dams aren't out yet. The dams have holes in them, and they're being removed, and there's actually still a lot of sediment to play out. There was a water monitoring data release from this week that came out yesterday, I believe, that actually showed that the water samples are definitely starting to signal spore concentrations in the water. So it'll be fascinating to see how it actually plays out this year. But the hypotheses were that the massive sediment was going to distribute over the basically blanket and suffocate a lot of the annelids. And we'll see if that ends up being true or not. There is extensive annelid monitoring that's happening right now to see if the sediment found its way to the annelids, or if there's kind of a juxtaposition in some spots and no overlap in others.